So, Introductory Neuroscience and Neuroinstrumentation, the Action Potential, Lecture 2. So, Hodgkin and Huxley, I keep talking about them. They are the high priests of neurophysiology, electrophysiology as it were. And you see on the right, Hodgkin is on the extreme right, and Andrew Huxley is on the left. And the apparatus they used, you have a cathode ray oscilloscope, you have an operating dissection microscope, and then things to infuse and electrodes to record, so on and so forth. So Alan Hodgkin, or Sir Alan Hodgkin and Sir Andrew Huxley, they found out how the action potentials are generated in the squid giant axon. And it was a remarkable tour de force when they did it, because though a lot of evidence had been accumulating, they were the first to prove the actual mechanisms, physiologists. So they used the voltage clamp technique, which was developed by Kenneth Cole at Woods Hole in Massachusetts, to find mechanisms of action potential generation in the squid giant axon. So this is one of the nice things about science, that people collaborate and are very generous and share their techniques, and advances occur. Axons and neurons have a threshold for the initialization of action potential. So their resting membrane potential is about minus 60 millivolts, and if it goes up, depolarizes, goes towards 0 to about minus 45, you have an action potential. So increasing the voltage from minus 60 to 0 produces a large transient flow of positive charges into the current. It's called invert current. This is followed by a sustained flow of positive charges out of the current, out of the cell, and it's called outward current. So voltage clamping experiments by Hodgkin and Huxley demonstrated that the inward current is due to sodium ions flowing into the cell, and the outward current is caused by potassium ions moving out of the cell. Okay. So these currents, the sodium current or the potassium current, typically they are denoted by I subscript Na for sodium current, and I subscript K for potassium current. Now they can be selectively blocked, and this blockade does not affect the other ionic flows. The block occurs at the sodium channel. There is a sensitive area. It's kind of a lock and key mechanism, and there is this chemical compound called tetrodotoxin, shortened to TTX. Now this is a very powerful poison. It's found in the Japanese puffer fish. It's a delicacy. They eat it, but they carefully remove the TTX before eating. But even so, every year many people die in Japan of TTX poisoning, because it blocks the sodium channels at microscopic concentrations, 10 to the minus 5 molar. All your sodium channels are blocked, and it's irreversible. So similarly with potassium, you have specific channel blockers for potassium. It only affects the potassium channels. It's called TEA, tetraethyl ammonium. So if you consider the plots on the right, so you have an invert current. You have look at the central thick line. You have an invert current and followed by an outward current. This is during the action potential. Now suppose you replace the sodium outside and make it sodium free seawater. You don't have any invert current, but you still have the outward current. Okay, and in this panel on the extreme right, the plot above is where you have a TTX blockade happening, and you have only the potassium currents, and these are the different command voltages. It's a voltage clamp, minus 15, 0, 15, 30, 45, 60. So you see it occurs later. When you block the potassium channels with TEA, you only have the sodium current, the invert current, and that's the panel below. And these are the command voltages of the voltage clamp, minus 15, 0, 15, 30, 45. And it's a very fast acute thing and comes back to normal pretty quickly. So using TTX, we can selectively isolate the potassium current and examine its voltage dependence. And similarly TEA, tetraethyl ammonium, we do the same thing and look at only the sodium channels because sodium currents, because we have blocked the potassium channels. So these channel blockers show some fundamental differences between sodium and potassium channels. The inward, okay, let's consider the potassium, the sodium currents which are below. So you have an inward current and it occurs fast, but it also rapidly inactivates, and it's transient. It inactivates even when the membrane potential is 0. The potassium currents, on the other hand, they take some time to occur and then they are long lasting, and that's why they're called delayed, and it's also called rectifier. Okay, delayed rectifier. When you hear of the word term delayed rectifier, you think of potassium currents. So the sodium current both activates and inactivates rapidly, while the potassium current only activates slowly, like so over here, slowly. Okay, and the potassium current, there's an interesting property. As long as the membrane potential is clamped at this particular voltage, it is activated. Okay, it keeps happening, and it does not inactivate, it is sustained. But the sodium channel, is very different. It activates and deactivates fast, and the interplay between these two processes causes the action potential. So at the time when they found this out, these physiological phenomena, we were not sure, they were not sure, nobody was sure of what is the cellular mechanisms, what happens, and the beauty of electrophysiology is you don't need to know to figure out what's happening, but if you want to go at the molecular level, you have to have some idea. So they speculated with some kind of enzyme related activity, and that fit in. Subsequently, we've with molecular biology and the tools of molecular biology and structural chemistry, protein structure, we have now found that the sodium channel, there's actually a channel, and it is sensitive to voltage, and it inactivates and activates, and it's on the membrane. Okay, so these channels have a voltage sensitive gate that opens with depolarization and closes with subsequent repolarization of the membrane potential. So this process of turning on and turning off the potassium current is known as activation and deactivation. Simple enough. But sodium channel is slightly different. It also exhibits voltage dependent activation and deactivation, but the sodium channels become inactive also despite maintained depolarization, unlike the potassium channels. Okay, and on the right you see the molecular structure, the postulated molecular structure of the sodium channels. It has 4 units, and there is a place where TTX acts, tetrodotoxin, and blocks it. And there's another blocker called saxitoxin, similar to tetrodotoxin, blocks it. And below is the actual conformational, the biochemical. This would be the secondary, tertiary, and quaternary structures of the sodium channel. So channel activation and inactivation. So the sodium channel not only activates and deactivates as mentioned earlier, but also exhibits a separate process called inactivation, whereby the channels become blocked even though they are activated. So removal of this inactivation is achieved by removal of depolarization and is a process known as de-inactivation. So just to make it a little more complex, the sodium channels possess 2 voltage sensitive processes. One is activation, deactivation, and the other one is inactivation, de-inactivation. Okay, so let's step back. Where is the action potential initiated generally? So if you remember the microscopic lecture on microscopic anatomy of the central nervous system, you have a cell. This is a pyramidal cell, and you have all the dendrites which are not shown over here, and then you have the axon going down, covered with myelin and stuff like that. Between the cell body, the soma, and the axon, there is a segment called IS, initial segment, and that has a very high density of sodium channels compared to the rest of the soma, and it is a very small compartment and it depolarizes easily. So generally action potentials are initiated in the initial segment. and this is an electron microscopic image of the initial segment, which is in kind of light red, and the hillock and the soma. The axon hillock is where the initial segment begins, and the initial segment ends where the axon with the myelination starts. Okay. So once a spike is initiated, about 30 to 50 mu down the axon from the cell body in cortical pyramidal cells, this then propagates. So it moves. Now it can move forward down the axon to the synaptic terminals where it causes release of neurotransmitter, or it can move backward, antidromic. So the first one is orthodromic, where it moves forward. The second process, where it moves backward, is antidromic, and it goes and into the cell body, into the cell dendrites, and there it can modulate intracellular processes. Okay. So refractory period. So immediately after an action potential, okay, for 1 millisecond during that period, the cell is refractory. It will not be able to fire another action potential. This is because of the inactivated sodium channels. They have to recover. And regardless of the amount of current you give, it will not be able to fire. So this is the absolute refractory period of an axon. Okay. The relative refractory period follows the absolute refractory period, and here it's due to the there's after hyperpolarization, the potassium channels act as a delayed rectifier. So there's a continued outward diffusion of potassium. So here it's relatively refractory in the sense that if you use the same stimulus threshold to get the AP, that will not fire. But if you increase the stimulus current, and you can force it to fire. So totally, the relative absolute refractory period is about 1 millisecond. The relative refractory period is about 2, 3, 4 milliseconds. It depends on the cell. But one is that this prevents electrical activity from reverberating, in a ensemble, in a network of neurons, because otherwise you could have reverberation occurring, and if you have reverberation, then you have epilepsy. So normally, these processes prevent uncontrolled positive feedback reverberation happening in the network. The other interesting thing is, if you look at the absolute refractory period where it cannot be fired, that gives us an upper estimate of what is the maximum firing rate of a neuron. It has to be limited by the absolute refractory period. So approximately over here, if it's 1 millisecond, and it would be the firing rate, absolute firing rate of a neuron would be anywhere from 500 to 1000 hertz, and that's it. It can't be higher than that. Okay. The speed of action potential propagation is affected by myelination. Okay. So axons can either be myelinated, where they have a covering of myelin, which is an insulation. We will study this in detail. Or it can be unmyelinated, where it just has the cell membrane, but there is no myelin. Okay. So sensory and motor neurons of the peripheral nervous system, they are myelinated by a special glial cell called Schwann cell. So this forms a spiral wrapping of multiple layers of its cell wall around the axon. And typically, small vertebrate axons, like for example pain, C fibers which observe pain, and invertebrate axons are not myelinated, whereas large vertebrate axons are often myelinated. Okay. We will get into the details of the advantages of myelination in just a little bit. So in the peripheral nervous system, that is the part of the nervous system outside the brain and the spinal cord, several Schwann cells wrap around the axon along its length and leave small gaps in between. They're called nodes of Ranvier. So here on the right, you have a Schwann cell, and it's wrapping its membrane around the axon. It keeps wrapping it, so you have multiple layers. Okay, and in between two different such wrappings, you have a node where the axon is exposed, and it's called the node of Ranvier. So these are all names of anatomists: Ranvier, Schwann, so on and so forth. In the central nervous system, instead of the Schwann cell, you have an oligodendrocyte, which is a kind of glial cell, and each oligodendrocyte typically ensheaths multiple axons. Schwann cell, only one axon in the periphery. oligodendrocyte, multiple axons. But otherwise it is the same purpose and function. It insulates the axon. Okay. So we will study this a little more detail in neuronal biophysics, but for now, this myelination of axon reduces its membrane capacitance by moving the electrical charge differences between inside and outside further apart, therefore reducing their influence on each other. And this significantly increases the passive length constant of the axon. Okay. We will, the length constant briefly is the distance where the electrical potential decreases to 1 to the eth of its initial value, that is 37% of its initial value. We will get into details more, but this increases the length constant. Okay. And here you have the details. A cross section of an axon with its myelin cell. The Schwann cell is with its myelination layers. The Schwann cell which produces the myelination is outside, and in between myelin layers you have this node. It's about 2 microns. It's a node of Ranvier. And typically the distance between, differs in different axons, but the internodal distance is anywhere from 300 to 2000 microns, which is 0.3 to 2 millimeters. Okay. So the sodium channels which allow the upstroke of the action potential, they are concentrated at the nodes. So the generation of action potential at each node causes depolarization of several adjacent nodes, and subsequently generation of an action potential with an internodal delay of only 20 microseconds. This is referred to as saltatory conduction. Okay. Demyelination of axons causes conduction failure of action potentials. Example, multiple sclerosis, where your latencies increase and finally it stops conducting. Or it could be it could be diet or genetic, and it could be post-viral infections called the Landry-Guillain-Barré syndrome, where you have demyelination occurring and loss of nerve function, conduction failure. So some characteristics of central nervous system neurons. So we talked mainly about the squid, but this, what happens in the squid as far as the action potential, resting membrane, all these are pretty much the same, if not very similar, in all the neurons, vertebrate, invertebrate, mammalian, human, studied so far. But the details vary. So in the human central nervous system, neurons exhibit a wide variety of electrophysiological properties. They have multiple conductances, not just sodium and potassium, you also have calcium. And you have different types of sodium, different types of potassium conductance, and different types of calcium conductances. Now in the squid we saw sodium currents are transient, but they can be transient, they can also be persistent. And likewise potassium currents, they vary a lot in their voltage sensitivity and kinetics in different cells in this human central nervous system. So neurons, talking of potassium, they have multiple subtypes of high threshold potassium currents, and there are low threshold potassium currents, and they generate bursts of action potentials. And after this you have, when you have hyperpolarization, that process of hyperpolarization activates ionic currents which are involved in rhythmic activity, like central pattern generators in the brainstem which control heart rate, control respiration, rhythmic activity. Okay. So thank you very much. In the next session we shall consider aspects of axonology and neuronal biophysics.